In this screencast, I'm going to show you how to analyze Bushman data published in Nature on February 18, 2010. So here's the plan. We will identify all sites where Archbishop Desmond Tutu is different from the reference sequenced human genome. We will find all Archbishop's SNPs that are new and not contained in dbSNP, and we will find exons that house these new SNPs. We're going to do this with Galaxy, so now I'm going to switch to my web browser. And here in the web browser, I will simply type usegalaxy.org. This is Galaxy, and the first step here is to get the data. For this, I'm going to go to Data Libraries, scroll down, click on Bushman, and the data set I will be using here called All SNPs in Personal Genomes, which is a summary of all SNPs found in all personal genomes published in this paper. If you click here, you will see an example few rows of this data set, plus you will see description of the columns. What we need to remember here is the column 4, because that contains the reference nucleotide in the sequenced human genome. And another thing we need to remember is this column 14, which contains nucleotides in Archbishop's genome. So column 4 and column 14. Now I'm going to scroll up, click on this triangle, and go to import this data set into my current history. It's imported. Now I click on Analyze Data, and here it is. It's a fairly large data set. It contains almost 11 million SNPs. And the first thing I'm going to do here is I'm going to filter it. To filter it, I will go to Filter and Sort, and I will click on Filter on Ambiguities. This is the tool which was specifically written for this project. And here's the expression that I'm going to use. Well, first of all, I'm going to restrict this data set to chromosome 22 only, just to save us some time, because this is a relatively small chromosome, and so we don't want really large data set for this demo. The next expression that I'm going to use is going to be that I want to find all sites where reference genome contained in column 4 is different from Archbishop's genome, which and these nucleotides are contained in column 14. And finally, I would like to exclude all sites in Archbishop genomes which contain Ns. So this is my expression. Let's go over this again. C1 equals chromosome 22 will only choose those rows where the first column equals chromosome 22. Second expression here will only fetch those rows where contents of column 4 is not equal to contents of column 14. And finally, this will not print any data sets where any rows where column 14 is equals to N. Now we need to change this to unknown. That's going to be our meaning of N. N in this data set means we don't know which nucleotide is there. And we'll click Execute. This is a large data set, so this operation probably will take a few minutes. Now it's finished, and we've gone down from 10 million SNPs to about 19,000, and those are the ones on chromosome 22, and these are the sites where Archbishop Desmond Tutu is different from the reference genome. But let's identify those from that set that are really new. In other words, they're not present in any of the SNP databases. So to do this, we first need to go and grab SNP datasets from UCSC table browser. For this, I'm going to click on Get Data, click on UCSC main. And here in UCSC main, I will choose Variation Repeats. I will choose SNPs, dbSNP 130. And I will restrict this search to chromosome 22. And here I just need to make sure that output format is BED, send output to Galaxy is checked, and I just click Get Output and send query to Galaxy. So right now what's happening is we are fetching coordinates of SNPs from UCSC to Galaxy. So in essence, what we want to do is we want to subtract these SNPs from this data set. To do this, I'm going to go to Operate on Genomic Intervals and click on Subtract. And here I will be subtracting the UCSC SNPs, or dbSNP 130 SNPs, from my dataset number 2. So once again, subtracting these known SNPs from my large list, from my list of Archbishop's to-do SNPs. So subtracting 3 out of 2, click execute, we'll create dataset number 4. I will go ahead and rename this dataset by clicking on the pencil icon and saying new SNPs. Okay, we have 1400 of them. These are new SNPs. Now we want to restrict that list of SNPs to those that are contained within exons. To do this, we once again need to obtain exons from UCSC. So we're going to go back, click on Get Data, click on UCSC main, change this to Genes and Gene Prediction Tracks, choose UCSC Genes Track, make sure chromosome 22 is here, then make sure this is bed, this is send output to Galaxy, get output. On this page I will choose exons and click Send Query to Galaxy. What's happening now is Galaxy is uploading coordinates of exons for human genome from UCSC. And all we need to do now is first I will rename this to exons so it looks better. And now I'm going to go back to operate on genomic intervals. I will click on join this time, and I will join exons with new SNPs. Now this is done. So we started with about 1400 exons. We ended up with only 54 regions. And now we can visualize those at UCSC Genome Browser by clicking at UCSC main here. This will open a new tab or window in your browser, and it will paint coordinates with these exons. So for example, we can zoom in here. And see that this is an exon in this gene, and that exon contains a new SNP, and this SNP is restricted to Archbishop Desmond Tutu's genome. This is just 1% of the Galaxy functionality. You can do much more with these tools. You can extract sequences, you can extract alignments, you can perform various types of analyses, and to see more of this functionality, look at other screencasts at the Galaxy site, and the URL for that site is galaxycast.org, or at the main Galaxy site at usegalaxy.org.